the first definition which I would like to introduce to you is the alleles, and in a very simple form, it has been written that it is a different forms of a gene. Now we know that the genes which are present are on the chromosomes are a stretch of the DNA. Now we also know that as we received or any diploid organisms has received one chromosome from the father side or the paternal side and one from the maternal side. So let's say this is one chromosome. I'm trying to draw here. So this is one chromosome and this is another chromosome. These chromosomes, one is received from the mother side and the other one is from the father side. These chromosomes are also called as homologous chromosomes. Now the gene which is been present over here, the other gene is present in the exactly at the same location. Let's say we define this with the letter capital A, then there is will be another capital A which is in exact position. So technically, if we have to define, there is two genes which has been present because of the two chromosomes in the diploid organism. There is a possibility that this gene A and there is a slight difference in a gene which is denoted by small letter small a. These are called as the alternative forms of a gene. Let's take an example. Let's say there is a plant which is tall and we define its genotype with the letters capital T and capital T. There is a plant, another plant which is short in length and we denote its genotypes between a small t and t. So we also trying to know what is a genotype and what is also a phenotype. So the phenotype is basically the physical appearance of a plant. Okay, let me show you in a moment what I meant here. So if you see, this is the Mendel which he used for his studies, the height of a plant. So this plant is tall. This plant is a short plant, a dwarf plant. Okay. Now coming back to my previous slide here. So we denote this is by genotype. So the physical appearance of it is called as phenotype, and with the symbol letters in genetics we use this as a genotype. Now uh, if these two plants is being crossed, let's say plant 1 and plant 2 is being crossed, the next generation which it's come is called as the first filial generation. Now this progeny or this offspring will receive one chromosome from this and one chromosome from this. We can define them this is a female plant or a male plant over there. So this offspring will receive a capital T and a small t over here. Now this particular individual, now if we have to define the chromosome, if we have to draw this chromosome again, and I'm trying to explain the concept of the alleles, and this is a position and this denotes is by capital T, and the exact position you have an alternate form which is called as a small t. It also leads to another definition which we call the capital T and T as an homozygous plants because they have the same types of the alleles, or a small t plant is also called as a homozygous. But this individual which is having two alternative forms of the genes or two different alleles if you want to say, then these are called as heterozygous. Okay. Now the another terminology which is often used is the character. Now these are basically the characteristics of an individual that is been transmitted from one generation to another generation, and it is often used as a synonyms for the trait. Now coming back, taking you forward, that these 7 characters which is being studied by Mendel. Okay, now let's say we are taking an examples of the plant height. I can change the color code so that it is visible. So plant height, and we define that this is the genotype of these two plants, and phenotypically it looks like this, tall and small or short. The progeny of these two cross of a plant, we also saw that this is a heterozygous individuals consist of the two different forms of the alleles over here, right? Now, all these characters, the phenotypes which you are mentioning, they are also called as the characters. Okay, and the two alternative forms, that is a tall and short, is often used by the terminology which is we call as a trait. Okay, so coming back here, so this is how we defines the alleles, and this is how we defines the characters. I've also introduced to you the concepts of the genotype and as well as the concepts of the phenotype, and I also defined to you the heterozygous and the homozygous individual. I've also introduced a concept which is called as the F1 generation and the F2 generation, which is the first filial generation and the second filial generation. That is the progeny of mating of an individual of the P generation, that is the parental generation, and examples which we took, that is P1 and P2, is called an F1 generation. Okay, like if a man who is been married to a woman, okay, and all the progenies or which is there, these will be called as the F1 generation. Like I can be considered as an F1 generation for my parents. Okay, that's the personal example I'm taking, but to make you understand. Okay, similarly, the offsprings which I am having, those will be called as a second generations, and this is a second filial generation. You often heard this also in that someone who is grandparents, those are the parental populations, and then there is the first generation is their children's, and then they have another children's or the grandchildren's, those will be called as F2 generation. It is as simple as that. Okay, now we also come across a terminology which is often used, which is called as a gamete. Now gamete is a mature reproductive cell that is specialized for a sexual fusion. Now if I took this as an example here, and let me first rub it so that you can able to understand that what I am trying to say, how we basically talk about the gametes. Now the gametes, like for example, there is a plant P1 and it has been crossed to plant P2 having a genotype, let's say capital T and small tt. Now to define, because this is a diploid individual, or you can say they have the two chromosomes over here, okay, like this. This is another chromosome for this particular individual, and they have these genes which is been present and denoted by this letter capital T and small t and T letters. Now during the process of the gametogenesis, okay, or which is basically depending upon the sexes will be called as spermatogenesis or the oogenesis processes, the total number of the chromosomes becomes half. Okay, so let's say as a human being, if we take an example, okay, now as a human being, if they have a 46 chromosomes, and let's say this is a female, and there is a, let's say another individual which is a male. Now during the, she will be producing the egg cells. Okay, the egg cells which it will be bearing, whatever the number of the egg cells she will be bearing, this will be reduced to half. So each of these egg cells will consist of 23 chromosomes, not 23 pairs. Okay, not 23 pairs of chromosomes, 23 chromosomes. Similarly, a man who will be producing the sperms, these sperms which will be, let's say we denote these sperms like this, these sperms will also consist of 23 chromosomes and not 23 pairs of the chromosome. That's how it is been made into half. And the process by which it is being made, this process is called as meiosis. Okay? The meiosis. So how basically it works? So we denote this by a symbol that any diploid organisms by 2n. In some places you will also find it is defined as 2c. So the 2n number is 46. And what is the n number then? The n number is the 23. So which is the haploid sets of the chromosome. This is called as the haploid sets of the chromosomes. And as a diploid, so we multiply 23 by 2, make it 46. But during the process of the meiosis, or in the gametogenesis in specifically, say this first things which is doing it occurs in the cell cycle, and in the cell cycle process, if we have to define, maybe a separate lecture I will use to describe meiosis, but to explain the principle of segregation, I have to give little bit background, and that's what I am trying to give it to you. So there is a G1, there is a S, there is a G2, and the actual process of mitosis or meiosis which is taking place. So in this process, that is the S phase, there is a duplication of the genome take place. Means if there is a 46 chromosome, so initially they will becomes 92. So this 2n number, because if you remember this, the n number here is the 23. So now it will become as a 4n. And after this 4n, it will be in the first division, which is meiosis, this which is called also reductive division, will be equally divided as 2n numbers, and from these 2n numbers they will be made as n, that is the haploid sets, and similarly this is also made as an haploid set, that is the n numbers. This is how the gametogenesis process work, and these sperms or the eggs which is harboring the half number of the chromosomes, these are called as the gametes. Now during the process of fertilization, so this is an egg cell, let's say, and this is your sperm. It comes and fuse, so it is having n number, this is having an n number, and then it's basically fused to make a 2n number or a zygote. This zygote further divides from 1 cell to 2 cell, 4 cell stage, and then to up to 100 cell stage to make it is a blastocyst. From this blastocyst, then it becomes the morula. Then from this morula, it's basically the process of embryogenesis starts. Then it gets implanted into the uterus of the mother. Then placenta starts coming, and that's how the individual basically works. What is worth to mention here, or one should take home lesson which I would say here, is that each individual for the eukaryotic systems, they receive 50% of their chromosomes from their mother and 50% of their chromosomes from their father side, or the maternal and as well as the paternal sides. I hope that clears my point. The next terminology which is we are already defined, that is gene. So I've already defined that these gene, but for the Mendelian genetics, it is also called as Mendelian factors. He named those as a factors because at the time of Mendel, he was not aware of the concept of gene, DNA. He is not known to them. So he told them that these factors which determines the characteristics of the organism, and then he used those symbols to represent them. Like for tall height, it is capital T, or say for the round shape of a seed, then he uses a symbol like capital R and small r and so on. So we will be talking about that in a moment. And the genotype, as I mentioned, this is the genetic constitution of an organism, and the phenotype is the physical appearance of the organism, which basically governed by the, so we can say the phenotype of an individual is governed, let's say it is phenotype, is by the genotype plus the contribution from the environment. Okay, now this is very important here, because whatever we are, it is not only because of the genes but also due to the environment. If a gene it does not express, then there will be a problem with that. Okay, let me give you a real life examples to make you understand. The peoples who lives in cold areas, okay, they often the vitamin D's has been supplemented in the milk over there, and the reason for that, because they've been not exposed to the sunlight. And it has been observed, the studies has been observed, that due to the vitamin D from a very early age and the weather conditions, their height is comparatively much higher as compared to the person who is in a warm region. However, there is a more details is been required, because again the height of in humans is a polygenic inheritance. It is not simple as that. But what I'm trying to make you understand, that there is a contribution from the environments does takes place, and it is mostly associated with the gene expression and the DNA methylation and so on. Okay, so maybe in some other lecture we will discuss. The essence of today's lecture is to make you aware of the genetics terminology. Okay, I hope it is so far so good. Let's move ahead here. So these are the characteristics or the characters which were studied by Mendel. We defined this in our previous lecture number 1 as well. Now you see here is, that is the two things has been mentioned. Okay, like say for example, a seed color has been mentioned here, or a seed shape, a yellow versus green, or a smooth versus wrinkle, or a height of a plant, which we just took, tall versus a short height of a plant. Now these are two contrasting forms of the phenotype. Now to understand this, we will use a cross, and this type of cross is called as a monohybrid cross. When we say monohybrid cross, we are interested in knowing only one of the characters at a time. Okay, what is a dihybrid cross? I will take this is in my lecture number 3. But right now, just focus upon what is a monohybrid cross. Let's say there is a one plant which pod color is green, and another plant which is having a pod color of yellow. The genotype of this particular plant we can define as capital G and capital G, like this. And say for the yellow, for the simplicity sakes, we refer to as a small g and small g, just to differentiate the genotypes of this individual. Phenotypically, they are green and yellow. Now, what Mendel actually did in one of his cross, that he took this as one of the plant, and then he crossed this plant, that is a green pod color of a plant with a yellow pod color of the plant. So we expect that is the next offsprings, that is the F1 offsprings. Okay, that is how a genetist wants to know that how it is, and Mendel was trying to understand. So to his surprise, he found that all the F1 plants were green pod color. Okay, green pod color of the plants he observed. Now he used certain letters to make it understand, and this is basically what we are trying and know these things, that is called as the process of the gametogenesis, which I explained to you in a moment ago. Now in genetics, if we want to say that this particular offspring has received 50% of the chromosome, so we are actually interested in knowing only one character or one trait over here, that is the green. So that's G will be received. Similarly, this particular will also parent will also contribute